This is lesson number 5 in the circulatory system, all about blood. Goals for today are to be able to describe the components of blood, including the shape, function, and origin of the different blood cells, the components and function of plasma, and the roles of antigens and antibodies in blood type. Now, blood is a connective tissue, and it is composed of several types of specialized cells suspended in a plasma matrix. It has 3 main functions in the body, which is transport of gases, nutrients, waste, and hormones, protection, its role in immunity and also in blood clotting, and regulation, helping our body to maintain homeostasis through regulating pH, water balance, and our temperature. The components of blood are the cell part, which are white blood cells, platelets, and red blood cells, and the plasma component. Now, plasma makes up about 55% of the blood volume, and it's the liquid part. Most of the plasma is composed of water, and the water in the plasma helps to maintain our blood volume, which we know is important in maintaining a healthy blood pressure. Water is also a universal solvent, and so it's the plasma that does lots of the transportation of molecules around the body. Nutrients, gases, hormones, all of those dissolve in water and can be transported through the plasma. Proteins make up about 7% of the plasma. There are clotting proteins like fibrinogen and prothrombin. There's albumin, which helps to regulate membrane transport, the movement of molecules across membranes, and also immunoglobulins, which are antibodies involved in blood type and in our immune system. And the remainder is dissolved materials, the things that the water was able to pick up. Salts, gases, nutrients, waste, hormones, vitamins, and ions are all dissolved in the plasma, make up the liquid part of our blood. The cellular components, sometimes called the formed elements, are the solid parts, and they make up 45% of our blood volume. The majority of that is the red blood cells, also called erythrocytes or RBCs, and their main function is in transport, specifically oxygen and carbon dioxide. The white blood cells, also called leukocytes or WBCs, have a function in immunity and make up a small proportion, between 1 and 4%. The number of white blood cells changes. If you have an infection, a doctor would expect to find a higher white blood cell count in your body. And if you're healthy, no infection or other issues, then the white blood cell count would be much lower. And making up the last little bit, less than 1% of the formed elements are the platelets. And these are actual fragments of cells that are involved in blood clotting. They're also called thrombocytes. Now, the red blood cells, the most abundant type of cell we find in our blood, they are fairly small, between 6 and 8 micrometers, and they are biconcave, which means that they have a bit of a dip in the middle on both sides. The reason for that dip is that there is no nuclei or organelles in a mature red blood cell. Because of that, the red blood cells don't live for very long and need to be constantly recycled. There are between 4 and 6 million red blood cells per microliter, so they are very common in our blood. Males typically have more red blood cells than females do, and the number of red blood cells we find in our blood is related to the oxygen concentration in the air. So people who live or athletes who train at high altitudes often have a higher count of red blood cells. We also know that there are athletes who choose to do blood doping, where they have a blood transfusion to try and add extra red blood cells to make their body more efficient during high cardiac activity athletic endeavors. And as we mentioned, the red blood cells, they're primarily functioning in the transportation of oxygen, but they do also carry carbon dioxide and hydrogen ions between the lungs and the tissues. Now, hemoglobin is a molecule inside our red blood cells. It's made out of 4 protein chains, 2 alpha and 2 beta folded proteins, and each chain contains a heme group which binds to the oxygen. The heme group is made of iron and some other molecules. In cool, neutral conditions, such as those we find in the lungs, the heme group binds weakly to oxygen. In warmer acidic conditions, like in the tissues at the capillary bed, the heme group lets go of that oxygen quite easily. And so the conditions of our lungs and our tissues help to support this transportation of oxygen, picking up oxygen in the lungs where the air is cooler and releasing it in the tissues where the blood is warmer. Now, red blood cells are made in the stem cells of our bones in the red bone marrow, particularly in the skull, the ribs, the vertebrae, and at the very ends of our long bones. As an infant, less than 1 year old, red blood cells are made throughout the body. But as we grow and mature and eventually stop growing, the red blood cells formation is limited to just the ends of our long bones and the bones of our core skeleton. The stem cells found there are continuously dividing to produce new red blood cells at a rate of about 2 million per second. There are several developmental stages. They do begin with a nucleus, but as they gain hemoglobin, they lose that nucleus and shrink in size to help them flow more easily through the narrow capillaries. When the blood oxygen levels in our body are low, the kidneys are signaled to produce a chemical called renal erythropoietic factor, and that, combined with some liver proteins, can stimulate extra red blood cell formation. As I mentioned, they live for only a very short period of time, about 4 months or 120 days. Once they get too old or are damaged, then macrophages in the liver break them down, and they are recycled by the bone marrow and spleen. Iron is also recovered from the hemoglobin and sent back to the bones for recycling. The heme portion, that protein portion, can be broken down and is part of what makes up the bile salts, the bilirubin, that get excreted from the liver and help in fat digestion. You can see the full life cycle of a red blood cell. Now, common disorders of the red blood cells are generally called anemia. Anemia is when there are a low number of red blood cells or deficiency in the hemoglobin. Sometimes this is also called iron deficiency, but that's actually only one type of anemia. It can be caused by diet, by blood loss, by vitamin deficiencies, or a number of genetic disorders that can also cause anemia. The white blood cells are much larger than the red blood cells, between 8 and 20 micrometers across. They do have a nucleus and appear white in color rather than the red color from the iron in our red blood cells. However, on a microscope slide, they stain purple or blue, and so you can see those in the picture here. There are far fewer white blood cells for red compared to the red blood cells, but 1 white blood cell for every 600 red blood cells. And our white blood cells are a vital part of our immune system. These are the cells that fight off infection by producing antibodies or by engulfing pathogens. They are also formed in the bone marrow and also in the lymphatic tissue. And white blood cells are not limited to the blood vessels. They actually can squeeze out through the membranes and attack invaders in the space between the cells around the blood vessels as needed. If you've ever had an injury or a cut and you've seen pus, that white material is actually white blood cells that have moved out to attack foreign bodies or pathogens in the space around your injury. There are a number of different types of white blood cells, and we won't ask you to memorize all of these, but here they are for completeness. The basophiles, which are white blood cells involved in a histamine release, which is something that can cause symptoms like allergies, but also causes fluid leaking into the tissues and swelling, which in the case of a foreign invader is useful in flushing out any bacteria or viruses that may be entering the body. The neutrophils, eosinophils, and monocytes are all white blood cells that phagocytose pathogens, which means they wrap around and engulf pathogens to destroy infections. And then the lymphocytes are the blood cells that are responsible for our specific immunity, the immunity that develops after an exposure to a pathogen or after exposure to a vaccine. So here, if you look at this video, you will see the phagocytosis of a bacteria by a white blood cell, specifically a neutrophil. They do have slightly different appearances. Again, we won't ask you to memorize or label these based on appearance, but here they are. And also in your textbook explained in much greater detail. Now, white blood cells are a common diagnostic tool. Elevated white blood cells counts can signal disease or infection, and the type of white blood cells also can give a hint about what type of infection might be present. So different types of white blood cells are common after a bacterial infection, after a viral infection, fungal, cancers, or other allergic reactions will trigger different types of white blood cells. And our antibodies and antigens. Now, these are antibodies are proteins produced by the white blood cell lymphocytes. Antibodies have receptors that bind with complementary proteins called antigens. Antibodies are used to recognize the antigens of foreign cells. Sometimes those foreign cells are pathogens, like a bacteria or a virus. Our body also produces antigens for self-recognition, so we know which cells belong to our body and then don't attack our own cells. Usually, white blood cells do not produce antibodies that match their own antigens. If they do, we would consider that to be an autoimmune disorder. Auto meaning self, and immunity being that attacking or the protection against foreign bodies. Blood type is another example where we see this antibody-antigen interaction. And so the ABO blood groups, there's type A, type B, type AB, and type O. And those are named based on the type of antigen found on the surface of the red blood cells. In blood type A, there is A antigen on the red blood cells, and that means that the plasma contains B antibodies. The B antibodies do not bind with the A antigens, and so no agglutination will occur. If you remember back in grade 10, you likely did a lab looking at these blood type groups. Now, the platelets are actually cellular fragments, so they're very small, between 2 and 3 micrometers, and they're the result of the breakdown of megakaryocytes. They are formed in the red bone marrow, just like the red blood cells and white blood cells, and they live in the bloodstream for very, very short periods of time, between 8 and 10 days. The job of the platelets is to form blood clots in response to an injury or tear in blood vessels. Platelets at the site of an injury grow sticky tentacles and send out chemical signals that attract other platelets and release clotting factors to form a clot or a scab. This stopping of bleeding is called hemostasis. Hemo meaning blood and stasis meaning stopping or stability. And so when a vessel is injured, you get a cut of some kind, the body seeks to maintain this hemostasis in 3 different ways. The first way is through vascular spasm. So the blood vessels leading to the site of the injury actually narrow and restrict blood flow to the site of the injury. So less blood is getting to the cut. The second thing your body does is platelets start to aggregate. So those tentacles grow and the sticky platelets stick to the wall and also stick to each other to start to form a plug. They also release chemicals which recruit more platelets and trigger what's called a coagulation cascade. And finally, we have final clot formation. Proteins are activated by the chemicals released from platelets, and they form a network of fibrin to seal the wound, and that's the actual scab or clot that forms. There are enzymes and hormones involved in clot formation. Those activated platelets with their sticky tentacles release an enzyme called thromboplastin. And in your notes, you can see some arrows and blank spaces to copy down this diagram. That thromboplastin enzyme activates a plasma protein called prothrombin and becomes thrombin. A coenzyme is needed here as well, the calcium ion. Now, thrombin has an activating effect on another plasma protein called fibrinogen. Fibrinogen becomes fibrin, and that's the actual protein network that forms the scab itself. It traps blood cells and stops the bleeding. So one more time, we've got the thromboplastin that is released. It activates prothrombin to become thrombin. Thrombin activates a fibrinogen, which is soluble. It dissolves and transports in the liquid plasma, and it becomes the insoluble fibrin, which is what forms the strands to trap and stop the blood flow.